Here's another question. What state does the sponge have to be in? Like, do we want it to be as whole as possible, or?
I mean, that's ideal, but
Okay.
even if we just get some of the liver looking bits, we can still barcode the DNA, so
Yeah,
if we get
that
smashed, it's
that was kind of
fine.
the idea, is that if we wanted to get more, we could also use the slurp.
Okay.
Yeah.
What are you going to try first?
I mean, well, so if you want, the slurp will be like the fastest, most efficient. We might have floaty bits come out of C. I believe there's floaty bits in C. And this will be also floaty. And putting into the AFD. So we might.
Plus, if we're going for a larger clump, we may have to make 2 passes at it. So
yeah,
I might recommend doing the slurp on slow.
Yeah.
Okay.
Keep it intact.
Yeah, I will definitely bow to your guys' judgment.
Let me get that camera set up.
Roger. Do we have anything in the slurps
camera?
Right now? The bucket. Do we have anything in
Sarah,
the
is there anything
slurps?
in the slurps?
No, they're empty.
Roger.
Has it been flushed?
Not that I have seen.
All right, so I'm going to start off by just running the flush.
Roger.
And Jess, I would touch the sponge with the arm, figure out if it's something that we're going to just slurp right into the slurp, or whether you're going to grab it and put it in front of the nozzle.
That's a good point.
And we'll bring it in. So
yeah.
I can run this while you do that.
Sure thing. We can try giving it a poke here. It looks like it's going to be pretty friable.
All right, slurp is running. We are flushed. Slurp is secure, and I will rotate your jar.
Roger. Little gangly arms here.
The wrong way, because forward is labeled backwards.
2.
And are we okay with getting some of the associates?
Yeah, that would be ideal.
Okay.
Go for a 2-fer. Or a 3-fer, maybe.
Yeah, bonus points if you get a shrimp. Shrimp are hard.
Greg, you have 1 lined up, right?
Yep.
Cool.
So you want some live bits, that's correct? Not the sedimented ones?
Yeah, I think ideally we would get more of the live, but...
All right, Marley, you want to go ahead and zoom in there a bit, please?
Yep.
So associates, tissue, live.
And you may have to get some like into the end of the nozzle and then kind of push it against a rock to cut it off.
Yeah, it looked relatively friable, but yeah, that might be a good tactic too. And a shrimp, bonus points for shrimp. That's great, Marley. All right, ready?
Yep, you're running 50%.
Roger.
Oh,
Come on, Shrimpy.
Shrimp doesn't want it.
All right, here you go, shrimp.
it reminds me of tissue paper.
Yeah, we got a shrimp.
no.
Oh,
No.
no.
He's tricky.
Evasive.
Do you want me to go for him one more time?
I don't know, I think he's kind of earned it.
Roger. Your call, not mine. Sam, are you watching up on 7 there? Let us know when you have satisfied the
Oh.
Do you see it coming in?
sample.
Yeah, it's really hard to judge how much that is, but...
I'd say it's probably like a large handful right now.
Okay, I think that's... Oh, nice. I think that's probably about good then.
All good?
I'll take a little bit more. We don't want to be shy, but we also don't want to take too much. So I guess kind of clean up your area.
Clean up your area.
This is sample 026?
026, correct.
Okay, great, thank you.
come on.
I can always increase the slurp, but just a little bit. But we've got a nice
We
clump.
got a lot now
Let
in
me
there, huh?
Bump us.
Oh yeah,
up.
that's a nice amount.
Come on. There we go.
Cool. Nice.
Good. Is that good for you, Sam?
Yep.
All right. Go ahead and stop suction there.
I'm
then.
gonna let that last bit come through.
All right, I'll give it a good shake.
It looks like a pillow exploded in
We had a pillow fight down
there.
something. All right. Marley, you want to go ahead and come wide there, please? Yep.
And yeah, shrimp are really hard to get. The best way to get a shrimp is
Get it from the back.
behind. Yep.
Yeah. We went on like a shrimp hunt once, me and Bob. Probably 10
I think it
minutes
was
trying to get a shrimp.
might have been the Galapagos, but what are they? The super tiny shrimp that are around corals. They're like smaller than the size of your fingernail.
I don't know.
Like the little copepods or krill or something?
No, they look like shrimp. It was something that sounded like mouse because I said I was going to call it.
Mysid.
Yeah.
Oh.
We
Subsea
they were oddly there was a group of them like they were schooling just in this one kind of pocket between a couple of rocks, and so we got them with the slurp by rotating the slurp back towards the camera and getting one between us and the slurp, and then because we only wanted to get like 6 was the limit. So we couldn't just like go in there and try to take everything.
That would be so hard with mysid.
Would you be able to turn the jars to
Yep,
flush?
I'm gonna go ahead and bring us to flush.
Nice.
And as we're flying, I'll run it to clear it.
Alright, I'm gonna
Okay,
put in the
sounds
ship
great.
move.
Yeah, let's do it. Last minute visuals on this? Are you guys good?
I think we got plenty.
Roger. Coming up.
Coming up. Yeah, Sam, that's why I was asking. If you're using it for DNA stuff, then like we can use the slurp and it's just faster.
Mo better.
Yeah, well, with how fragile that was, I don't think we were going to get an intact chunk anyhow, anyway we did it.
Sponges are usually pretty difficult.
Yeah.
So here's a good rock
Mm-hmm.
coming up there. 40 meters.
All right, I'm gonna run your slurp.
Roger. .040, is that correct? Roger. You want to take a look at this?
We could do a zoom on it, yeah.
Quick Sam, Roger.
All right, that seems happy. Slurp secured. I'm gonna switch your cameras back.
Roger.
All right, you've got both rail cameras.
Thank you. All right, video, go ahead and push on in there, please. Let me know when you have had enough of this coral, Sam.
Yeah, thanks. Could we do, is that all the zoom we have, or?
Yeah, that's all the way in.
Okay, thanks.
Did you want any more tighter on the palm so I can go forward to it?
No, it's okay. I think we've seen this guy before.
Roger.
That's good on this guy.
All right, go ahead and come wide there, Molly.
Thanks.
Coming up.
So I know we're trying to make up some time. Where are we trying to head to? Are we just trying to catch up to our next waypoint or to a certain depth?
There's just a transect specified to go over this, to ascend up this feature. So we're just trying to make sure that we can do that in the time allotted for the dive. So we're just continuing to ascend this along the Baker Island.
Do we have a ship moving right now, or is it?
Yeah, we have a ship moving right now.
Alright.
Are we more or less headed towards waypoint 3 right now?
Yes, we are.
Okay.
I think this is from, we went back and we're
Yeah, we
looking
went back
around
10 meters for that
Yeah.
sample.
for that
That's right.
sponge.
That's right. Thanks, Ben.
Yeah.
And Craig.
Could we get a quick close-up on that blue,
The
maybe
blue.
coral on the rock there?
Roger. Lower screen.
Well, that's neat.
Good eyes. Doing it from the side so we can see more of the fan.
Yeah, that would be great.
Sure. So there's a piece of it on the ground there.
Yeah, it looks like you can tell where it broke off.
Yeah. All right, video, go ahead and start pushing in there.
Is that Victor Gorgie?
Looks like it, yeah. Yep.
So is that a like gorgonid sponge?
This is an octocoral.
An octocoral.
Yep.
Gorgonid coral.
We got an
Nice.
anemone.
Sorry, I'm not very stable here.
Oh, that's right. That's good on that.
Roger, go ahead and come with him, Marley. Anyone want some? Come on, George, you know you want some. Right. You want to zoom on these corals while we have the time?
Yeah, maybe especially that one on the right more.
The more golden looking one?
The one on the bottom.
Sure thing.
I'm not great with colors.
No worries. Me neither. All right, Marley, go ahead and push on in there.
Another crested gorgidae. Little squat lobster hanging out too. I think both of these we saw on our watch had a squat lobster hanging out in the middle too. It's interesting.
Not a bad place to be.
Yeah, I think one of the chrysogorgids has like a,
You go ahead and push full in there if you want, Marley.
like there's one species
there's
of
one
squat lobster
below
that's
it,
always
too.
on one species of, I think it's Metallogorgia. And they think they just like grow up together basically.
Aw.
And it's like always like that squat lobster on that coral.
Oh,
That squat lobster below doesn't have a coral.
Oh,
poor baby.
just keep waiting, buddy.
Keep waiting.
Looks like he has different front claws, though, right? The one below is a bit hairier
Yeah,
front
they're-
claws.
Yeah.
So we're good, but this is also a nice highlight. So if we want to take a few more seconds, that's
Sure thing.
good with me.
There's some of those mice
Are you
that
stable?
around.
Yeah, I'm stable right now.
Marley, you want to push all the way in and get a,
I think I'm all the way.
you are all the way in?
Yeah, I'm all the way
Alright.
in.
Roger. Nice. Gonna do a snap zoom on this one.
Sure.
All right. We have a little bit of time. All right, Marley, I'm going to ask you to push back in there.
Okay. Do you have the camera the way you want it?
Yeah, right there.
There we go.
We might need to do this one a little bit quick because the ship's moving. Feel one motion sickness bump up. There you go. Maybe you can try flowing on that one real quick.
Nice. That's probably good, thanks.
All right, that's all
Roger.
the way.
All right, Marley, you want to go ahead and come full wide, please? I need a boogie. Nice, thank you. Just here and we'll go. Coming up.
I'm doing 30 meter. Oh, yeah, 30 meter.
Oh.
Fan.
Do you want to look at that coral to the left?
You guys want that one?
Oh, sure. Thanks, sir.
I'll do it really quick. I'm also a little bit behind, so after this one I'll boogie up the
Yep.
slope.
Just make it a quick one.
All right, Marley, you want to push in there real quick? That's great. See you, George.
See you, George.
All right, you want to come wide, please?
That's good, thanks.
Yeah, I got a boogie. Sorry, guys.
No worries.
Coming up. That was a pretty one. Let me know if there's one that you guys want to zoom on, or else I'm gonna get in front of the ship.
Oh, sorry, I was muted. That's okay, we can keep moving.
Better. An enemy. Oh, it's cute.
So a lot more of them now. Is that a function of the depth that we're at, or is that just pure chance?
Well, different species will definitely have different depth delineations. Sometimes it's common to start seeing a little bit higher abundance when you get a little shallower. Food supply gets a little better.
Alright, we're at a more comfortable spot. So if you guys see one that you want to zoom on, let me know.
Yeah, we could stop and zoom on one of these.
Roger.
Doesn't matter which.
Okay. You're fine, Ben. I'll go the one over here. So there's some crinoids on top. All right, Marley, go ahead and start your zoom in there. That's great right there, I think.
It's a nice shot of the crinoid.
Jess, can we turn the lasers off just for a second? I think this might be a nice highlight shot.
Sure. So I fly a little more stable. You back up a bit.
That's all right.
Is there a little slug on the top there? Just above that uppermost crinoid?
All right, another few more moments there, and then we're gonna have to go. All right, Marley, you wanna go ahead and actually come wide? We're gonna catch up to the ship now. Yeah, go ahead and come wide there.
The creature?
Yeah, what is that?
Yeah, the kind of translucent. I don't know. At first I thought like they were shrimp or squat lobsters, but the
I mean,
one on
that
the
one
top.
is a
Yeah.
squat lobster.
Yeah, but the one right on top, yeah.
I don't know what that is.
All right, we can do a quick snap zoom
An
as
alien.
far as you can go, and then
Oh, maybe it's a homalid crab.
It's got legs, yeah.
Humbolid
It's a
crab.
homalid crab, so it has like a sponge on its back.
Oh, wow.
So we're looking at it from the
Marley, you wanna
behind.
push all
Trying
the
to
way
Holly,
down
did you
there?
hear
Time
that?
to go.
There's a homalid crab with a sponge on its back.
Marley, you wanna go
It has
ahead? Are you all the way?
it's a crab with a backpack.
I knew Holly was going to freak out about it.
We gotta pull away, I think, but
It's pretty exciting.
maybe we'll see one. Ooh, we have a lot of corals here.
Yeah, we're
I
gonna have to
can-
catch up to the ship.
Okay.
Or unless you guys want to stay
If
here.
you guys want to stay here, I can bring the ship back.
No, well, let's move forward.
All right.
We're still a little behind on ground to cover, so.
Yeah, but so homalid crabs have like modified hind legs where they can hold something on their back as like, we think it's for protection in some way. And so it was holding a sponge.
Like a little sponge backpack?
Yeah.
Why might it be carrying a sponge?
It's thought like to be some type of protection for them. So like, cause they're crawling around kind of exposed. But sometimes it's coral, sometimes it's sponges.
Oh, really?
Yeah.
So they carry other stuff.
Yeah.
Sometimes it's an enemy's, correct?
Yeah.
Yeah,
But the anemone ones aren't homolids. They actually use the anemone as like a shell, like a hermit crab would.
Huh.
So, like protection, so something that would maybe eat the crab will instead grab the sponge.
Yeah, and like the sponge
And
has
you wanna hold off
spicules
on
in
that
it, so
stuff.
things
yeah.
don't
Jess,
want
I
to
would
eat the
just power
sponge.
forward.
Right.
Or there's
Jess,
some thought that like maybe
I would just
there's
power forward.
chemical signals that
You
like
know,
come
run
off
your
of the
vert-
I
run
think
your
that
that are
is
different
horizontals
than
at
if it was
full.
just a crab.
I think
Mhm.
there's
But-
I think there's already a step that's kind of finishing
Not
up there right now.
totally sure.
And we have a wall coming
I
up.
also just had my arms behind my
Jess,
back
can
for
you
like
move
a
your
minute,
microphone
pretending
up
to be a
just a little
whole
bit?
You were physically like showing me your modified
my modified limbs.
limbs.
Coming up.
Wow, this is pretty steep.
Ooh, yeah, we're ascending the volcano,
That sounds cool.
right?
on the last cruise there was a crab. I don't know what it was, but it, definitely not a homolid because it had an anemone that it was
Yeah.
carrying, and it was like a bright pink one with like long tentacles. It just looked
Cool.
like it was going to a disco or something. It was like really.
Was its body like in the anemone? Did it look like it was like using the anemone as a shell?
Maybe, yeah.
I think they're called pagurid crabs
Pagurid.
or something like that. Let me see.
Those are the same as hermit crabs, I think. Just
Yeah.
regular old hermit
Yeah,
crabs are percurids.
Really? While you're looking up that crab, so up on Baker Island and Howland Island,
Already
they
fit.
have a huge colony, or a huge, you don't really wouldn't call them a colony, but a lot of strawberry hermit crabs, like thousands upon thousands of strawberry hermit crabs live on this island. And so there are these two historic day beacons that are pretty much the only structure left on there. And so during the day, the crabs will like try to find whatever shade they can. And so the interior of the beacon is just full of hundreds and hundreds of crabs, of strawberry hermit crabs.
Oh my
It's,
god.
a little disturbing. Oh, hey.
Yeah, there's a couple. Passed over a couple corals.
Yeah, the next one we can zoom in on.
Yeah,
We're
let's
just
get you out farther in front.
of
Yeah,
the-
yeah, I understand.
But we have the slope flattening out in front of us pretty soon.
There's one, when we see one that's kind of like what we're passing over now, if we see another one of those.
Okay.
Oh, there's a very large sea cucumber.
That is.
Some dead sponges on
Alright,
that rock.
pretty far ahead out now. So yeah, next one we see, we can zoom.
Sounds good.
Is that one?
I think that's
Yeah.
the one that we were seeing
It's,
before.
I don't think that's the one that they want us to see, but
Yeah, that's, I think you guys have seen a couple of the ones that are right below us. Did you see some of those when Sam was in here?
I think so.
Okay.
Yeah.
The one that's just below center right now, it's like kind of a yellowish.
Yeah.
Can we take a look at that?
Sure thing.
Oh, and there's a hemicorallium, that pink one over there too.
Is that the bubblegum coral?
No, it's a Corallidae is
Oh, okay.
the precious coral is the group that belongs to.
Alright, video, go ahead and start pushing in there, please. Marley, go ahead and start pushing in, please. Looks quite not happy, huh?
Yeah, it looks weird.
Mm-hmm.
Sedimented?
I think it's a black. Yeah, can we get closer?
Yeah, okay. Marley, you want to come a little wide and I'll
Yeah,
sit down? That's great. So I can see. if that's enough light for you. Go ahead.
Yeah, it looks like a black coral. It's just got a lot of sediment on it. So like that sponge earlier, the sedimentation rate is just kind of, I mean, suffocating is a strong word, but that's kind of what it does over time. They're not able to feed because they just get too much sediment stuck on them.
So there are definitely different corals that do
Alright,
better
well, you
at
can go
different
ahead and push it more
sedimentation
if you
rates.
have any more
Yeah, I
to.
think so. So like these black corals are have a lot of mucus.
You can go ahead and push it
So
more if
it's
you want,
probably
Riley.
that more of the sediment is sticking to them
Right.
than like the.
Oh, you can zoom in more if you want, Riley.
Okay.
Sorry, I think my SPL is a little up.
Yeah, you keep moving your mic up, and as soon as you let go, it drops back down.
Yeah.
Little hermit crab on there.
Oh, that is, yeah, you can see his little shell.
Yeah.
Did you also want to look at that? Did you also want to look at that precious coral? Or you, like, just
Yeah,
tape it
could
here?
we? That's good for this one.
All right, go ahead and come wide, Marley.
You just have to speak with your announcing voice.
I know. I need to get an announcing voice. Oh. Is that one up there?
I think it's just to the right.
Roger.
Are we?
Alright.
Yeah, sorry.
No, it's fine. We got the scope. We got the time. Just to the right.
More dead sponges.
Yeah.
Oh, it's the one on top of the rock there?
Right here.
Ah, roger. Okay.
The bubblegum coral?
I think it's a corallid, like a corallidae.
Not a bubblegum.
Not a bubblegum.
That's what I thought too.
Still cool.
Excuse me, Mr. Rock. See if we can reach the lights there. Let me know if that's too far away. Go ahead and start your zoom in there. Maybe it's too far.
So
Is that a
squall
squall
lobster.
lobster?
Yeah. So the white
That
part
looks like
is
it
just
broke off of
right.
something, and it's
Yeah.
just dying.
Does
I mean,
it?
Would
Does
that
it look
be
like
the
it?
stalk above us? There's sort
Oh
of that
right,
broken
there's not
Yeah.
really
stalk
like
up
a
there.
base. There's not like a. There goes a scale worm.
Yeah, it looks like it lost a lot of soft
Yeah.
tissue.
I wonder where it came from.
Not,
That base looks like it has something sticking out of it.
Well, but
but
it's
not-
broken.
It's broken up right
Yeah.
above.
Did you want to look up? Ooh, thank you.
Is it thick enough?
I don't know.
The stem
But it-
coming out of the base looks thinner than the coral.
Since this is already looks like it is fallen over, could we sample this? We don't have to take the whole thing, but it's pretty brittle. So if it's already fallen over, then it's not a huge deal if we break a piece off.
Sure, if that's what you guys want. We can do anything you want. Marley, you want to go ahead and come line? Do we have any ship
No,
together?
we're stopped right now.
Cool.
You want me to come full wide?
Yeah, full wide would be great.
Maybe it came off of that one that's up top.
Hmm.
There was a piece of live, but it's not very big up there. So maybe it
Did you want to look up?
snapped off.
That one up there, you mean?
Oh.
Yeah, I don't know.
Huh.
Definitely came from somewhere because there's no big base. There's no like base grown onto the rock,
Yeah.
like how we see with that dead one right next to it.
Yeah. So you want this one?
Yeah.
Roger. Get myself a new seat.
Megan, do you know if the Niskin that was taken earlier, was it considered a high biodiversity spot?
Let's take a look. One
Or
second.
was it like the control?
Yeah, it was near a Caligorgia coral and some associates, so
Okay.
it wasn't a control for sure.
Okay. Cool. Thank you.
Hmm.
Okay. You have any more tighter shots on this before we take a sample?
I think we're good. We were in on it pretty tight.
Roger. Oh, sorry, Greg. Good catch. Justin would be mad. Right. Marley, you want to push in there a little bit, please?
Yep.
That should be good right there. All right, Alexis, so tell me what you want and how you want it.
Yeah, so is it easier to come from the top of it if we want to take a couple branches?
Come from the top of it?
It's like the closest to the laser side. So if you came in and like put your jaws around like your cutting edge.
I see, okay. So you're thinking more something like this?
It's not giant, so if we...
You'd also take
Would this fit in a bio box?
It might fit in a big one. We still have E on the starboard side and B in the forward that are open, so
Let's just try to
scoop it up.
Scoop it up. It'll
Okay.
probably break some of it off anyways, and then we can just go into the big bio box.
Okay. Whole body. Oh, yeah.
It's pretty little.
Pretty little, yeah.
It's deceptive. I thought it was huge.
I know, I thought it was bigger too. I mean, we have lasers, we should know.
Yeah, it's true.
Maybe go back here.
Yeah, that's why I
Oop.
opted for one that's already broken off,
Yeah.
because then we don't have to worry about the rest of the colony.
It's so dense for being so small.
Mm-hmm.
With this type of fragmentation, is this important for their reproduction and for deep sea corals as well?
More so for scleractinians, so they fragment a lot in order to keep growing, but this likely would, over time, just the polyps would die.
Right.
It might be able to reconnect to a rock, but I think the sedimentation right here is too high.
So I would just go for the forward B box. So forward right box.
Forward, right? Roger.
Yeah.
That's great. Yeah, Marley, you want to come full wide, please?
Yeah.
Forward right box. Oh, stay.
We're losing.
Should be okay. That's a pretty long one.
Okay.
And we have 3 of those.
Slam dunk. I'll actually put my jaws in here a little bit.
Great.
That was awesome.
Yeah, well done.
That was fun.
Jess, you're getting a lot of praise online for your
Yeah, that was great.
Soft
Jess.
touch there.
Yep.
Cool. Well, glad people like basketball. Sounds good.
Jess, you were talking last dive about how your work here as an ROV pilot really sort of influenced your own research in terms of like studying a delicate grip and then looking at biomimicry to see how other organisms do that and how it can apply to your work. I thought that was really fascinating.
Yeah, it is a pretty
So
interesting
might be
question.
where it broke off.
Maybe,
I think.
yeah.
Did you guys want to look at it real quick?
It's okay.
Roger.
It kind of looks like on the left of it, there's a little branch that's missing.
Yeah.
Yeah.
Oh, here's
Yeah,
another one of
there's-
those black corals. And there's a,
Do you want a quick zoom on the black coral?
yeah. I want to see if this one has sediment on it too.
Sure thing.
They like blend in with the rock really well.
Yeah.
Because they're
It's
covered in the same color
camouflage.
sediment that's all over the rock.
No, I think their tissue is also like
Oh,
kind of
okay.
that color.
That too.
Yeah.
Okay, go ahead and push it in there, Marley.
Their tissue is sediment colored and their skeleton is black colored. Just
It was
like
just like,
the sediment
Yes.
and
Yeah.
black
Looks like
rock.
it's still pretty heavily sedimented.
say that again?
It looks like it's still pretty heavily sedimented.
Yeah. Yeah, I think it's just the tissue type.
Mm. Yeah, I
Thank you. That's good.
Sure thing. Go ahead and come line, Marley.
Little shrimp.
All right. I'll start to jet.
What do you think that little
Numbers.
blob was?
Oh no.
Yeah.
Little blob up there? Because that looks like a sponge, no?
Oh, that one does, yeah.
Oh, yeah.
No, the
There was a little red blob on
red
the
blob on the black coral.
Yeah, I'm not sure.
Blob on the black coral.
Red blob on the black coral.
Wait, was it you and I talking about what we're gonna nickname dives? Oh, shoot, maybe somebody else. Because we saw a lot of blobs on this. Oh, it was Sam.
Oh, yeah,
Sam,
no, it wasn't me.
Sam and I were talking about blobs earlier. Because the first thing they saw, or like started zooming on, were some of the tunicates that were attached to rocks.
The predatory tunicates?
Yeah.
Oh, nice.
And here it comes.
Holly, getting back to your question earlier, yeah, so the thing that is kind of inspiring about being out here is you can ask yourself many questions when you're on these dives. You know, how do you, these grippers, or this manipulator that we have has the capability of, I think, 500 pound force of grip force. I think it's either 500 or 300, so that's not very delicate. I mean, it's really good for oil and gas applications and a lot of industrial applications, but when you're sampling, it takes a lot more kind of finesse and patience, and so it becomes an interesting question of how to delicately grip things, and a lot of organisms have developed really interesting mechanisms of gripping surfaces, whether it's rocky surfaces, rough surfaces, or soft surfaces. So when I'm looking around here too on the screen, sometimes it's curious to look for attachment mechanisms that are being used, or attachment devices that are being used by organisms, and you can learn a lot from those structures. So, yeah.
Yesterday we had a very excited Jess in the sampling, in the wet lab where we were doing sampling, because we had a rock
they did
pen.
find you for that.
Oh yeah.
Oh yeah. Oh yeah.
I'm gonna steal bubble.
Go ahead.
So we had collected a sea pen that uses essentially a suction cup to attach to rocks. And Jess was very interested in that mechanism that the animal can use to do that. So she came down and took some photos, examined it herself so she can apply some of that to her own research.
Yeah, and also just to geek out, you know. So
Yeah. It's another black coral, another primnoid.
I know we see a lot of examples of that in, you know, advancing robotics, right? I think about if you guys have ever seen the Gecko bot, I mean, these grippy hands allowing it to kind of traverse much more, you know, extreme terrain.
Yeah. Definitely, yeah. They had some interesting gecko-inspired adhesives they put onto grippers for manipulating objects in microgravity, which is pretty interesting. So you have like large objects. I think it was actually at JPL I was doing this. Aaron Parnas. So it is curious. I mean, that's all basically electrostatics and van der Waals based adhesion, these gecko grippers. The applications are pretty limitless once you begin to understand the mechanisms behind it.
Yeah, and much like we're doing here, if you could have some sort of gecko bot crawling outside of a vehicle in microgravity, much better than having to send someone else to go out there and do it themselves.
Yeah. Yeah, exactly.
There's a little shrimp with very long antenna.
Yeah.
Oh yeah, what a cutie. He's like, hey. You want to do a slight zoom if he comes into frame? Marley, go ahead and zoom in on this little dude if you want.
What's up, little dude?
Look at those legs churning.
Oop.
Yep.
Probably.
The never-ending treadmill.
Jealous.
Not jealous.
Not jealous. Is that Marley? Were you
Yeah.
jealous?
All right, go ahead and come wide, Marley.
Can't trust a person that prefers an orange to a donut. So.
Yeah. We are not sticking with the donut theme. Not having that.
Well, donuts are kind of sticky.
They
Lord, here
are.
we go.
Oh,
Yeah.
I see the pun you put down there, Greg.
So we are seeing a lot of those same black corals. When we see one that looks like we could do a collection, I would like to do that.
Sure.
How much
Yeah,
do we
you
have
tell
left
us
in
when.
this ship move?
I think about 10 meters, maybe a little less.
Looks like we have
Another
a
little
spon-
glass sponge.
Yeah.
All right, Jess, your
Probably
call.
a sea star in there, that little
Roger.
white
We'll probably find one,
Oh, yeah.
like a
blob.
further up slope.
This is a cool geological feature, too.
It is. There's
I
another
was
way
sea
just
star.
thinking
This
that
works is we'll
too.
never see one ever again.
I know, right?
Oh, there's one.
So
Pretty easy.
the black coral that you want to collect,
Mhm.
the just basically one with the sediment.
Upper
Yeah.
screen.
Well, it looks like there's a lot of them, so
Yeah.
I just want to get a representative.
Yeah.
Oh, there's two.
Yeah, does this one here work for you?
Yeah,
Alexis?
that's great. It's just
Good eye, Greg. Yeah, there are two. Nice.
Ooh, there's some squatties in there.
Oh, yeah. Was that a brittle star too?
Maybe. Let's see.
Or is that just the squat lobster's legs?
It looks like a pretty big squat lobster.
Yeah.
Science, how much of this are we getting?
Just a branch.
All of it?
Not all of it.
No, like all of the branch.
Oh, sure. 15
Can I zoom, Jess?
centimeters.
15 centimeters.
yeah.
Okay.
I'd love to do a zoom on it before we
Yeah, am I
sample.
good to zoom,
Sure
Jess?
thing. Yeah, go ahead, Marley. You want the lasers in it, or can I move the lasers off of it?
You can take the lasers off.
Roger. Just to make it a little bit easier.
Yeah, there's a
to zoom.
couple of shrimp in there.
Mm-hmm.
Brittle star. There's definitely a couple of squat lobsters. There's a little hermit crab.
Oh! Oh, that hermit crab's a Very
Oh,
adorable.
sorry, I think I'm moving a little bit. I'll resend her up. I'll reset myself once we get these images.
We'll just go for whichever branch you guys would like. Don't worry about associates.
That's a cute shrimp there,
Ooh.
Marley.
That
Shrimpy.
is
That was a
a,
nice shot.
yep.
Sharon, where do you guys
Oh
want to
man,
put
where's
this?
Paul?
Do you have a spot you
Different
want?
Paul.
Yeah. It could either go in the starboard D, so the tiny one in the aft,
Okay.
or the starboard E, which is the larger one forward.
How flexible is this? I remember it's pretty flexible.
Yeah, they're pretty flexible. Probably want to twist the wrist once it's to try and snap the
Yep.
skeleton.
I was thinking more about
Or shoving into D.
no, putting it into bucket 2. Save D for something else.
Oh, that's a good point. To put it in a slurp, you mean?
Is this good for imagery, Alexis? Do you need
Yeah, that was great.
Yeah.
Ben, can we do that step back?
Yeah, sure thing.
Do you want me to come full wide, Jess?
Yeah, go ahead, please. Yeah, Megan, do you want to do a slurp with this? Or do we want to...
We can slurp it. Yeah.
We'd snip it first and
Yeah.
then just
Yeah,
feed it into the
if you want to
slurp.
do that, that works. We only have one slurp. It's in 1, so 2
Yep.
through 7 are free.
Okay, that's a good idea, Greg.
It's a good
I'll
idea
just hold it in
too,
front of
because
the
these
pipe
ones
there.
are a little light.
Sure thing.
I'm going to run this slurp. Switch cameras.
Get my bouquet ready. All right, Marley, you want to go ahead and push on in there a bit? That's great right there. All right, Alexis, you tell me what you want. Any preference?
Honestly, any branch, whichever branch is easiest for you to get your jaws on.
Sure. I think the vehicle's not as stable as they should have it.
Megan, did you guys pre-label the slurp jars?
They're all labeled.
The 008 SL2?
Oh,
Might be left on.
Somebody might have left that on. Yeah.
All right, is this about good for you there, Alexis?
Yeah.
All right, see what we get.
Yep, there you go. Then you're going to want to rotate the wrist to try and get it to
Snap off, yeah.
actually break.
Think we got it. Nope.
Shrimp are hanging on for dear life.
There you go.
I think that was the last.
Now might be a good time to go, Shrimpy. Yeah.
Or most of that end up pulling away from you.
Yeah, it looks like we only got a little bit. We
No,
can
that's
maybe
great.
go back for another hair
That's
shave.
good.
All right.
You got
Let's
a
take
couple.
a picture of this real quick.
Definitely mucusy.
Yeah, look at that.
It
It's
is.
snotting out there. All right.
Is that mucus from the coral itself?
Yeah.
Marley, you want to come full wide, please?
This is the one that is, you said is stickier.
Yeah.
Okay.
Does that present any challenges around, sampling at depth? I know there are some things you can and cannot bring back up, but do you have to make any of those judgments on coral species as well?
No, they're generally all fine coming to the surface. Some of them, if
Marley,
we're
wanna
trying to
zoom in,
keep stuff
please?
alive, some of them are not
Marley, you want to push in, please?
good candidates for that. But for like doing where we're going to preserve them at the surface, they're all fine.
Yeah, a little more. That's good right there.
That good?
Thank you. Yeah, let's see if I can. I had to jump up a little bit. I'm just gonna come up.
Okay. Look like you got plenty of room underneath.
Think so? I think I'm gonna hit the rock. Oh yeah, Greg, yeah, you're right.
Ready? I'm gonna start.
Sure. Let me get a different angle because otherwise.
Oh, it's a little fish.
Little fishy down there.
Might get another
Run away,
twofer.
little fish.
No, stay.
No, it's gonna get slurped.
No, it's like it's slurped in
yeah.
there. We do
Yeah,
not
there
want
you
a
go,
fish
buddy.
Yep,
sample.
so
Oh my
that's
gosh.
starting to go in.
Cutie.
So if you let go,
Yeah.
Great.
Nice.
there it goes.
Nice. Well done. There it goes. There it
Not
is.
pull weight. Stay.
Coral. And a
Great.
slurp.
Check.
All right, go ahead and come wind them.
Almost got a non-associate.
Stamina.
Yeah.
There
Look
he
at
is.
him.
There.
Hi.
There you are, little fishy.
Oh my gosh. Can we?
Yeah, if you want to look at that,
Can we
I
look
can.
at him?
I can put it away on the bubble if you want. All right, Marley, go ahead and push on.
Here, I'll
into
dry
that
this
fish.
so you can take care of the arm.
Oh,
Ah.
oh.
Yeah, I'll just look over here.
Oh, it's so cute.
Well, there
There
was
he
a
goes.
sea star up there too. That's a really interesting,
Yeah.
like swim
Swimming,
motion
yeah.
there.
Really using the hips.
I love it.
Using the hips.
Bye.
It's like a little kayaker.
It was very kayak motion.
Oh,
Where are we?
It's just
so
moving all over the place.
he's gone.
Quit looking at yourself.
Meta shot.
That was very meta. It was a selfie. I just took an ROV selfie.
We have done that with GoPros
Yeah,
before.
we set down a GoPro
Yeah.
Oh.
on a stick and set it out there and drove away from it.
Meta shot.
When we did it before, we all like set it out and then everybody in the control van waved, but that made no sense because we weren't in the vehicle.
You do, because it's so dark.
You
and,
feel like you're, like,
you feel like you're in
yeah.
kind of a bunker. You do get the sense that you're actually there.
Yeah. But there's nothing quite like going there.
Yeah, I'm sure.
All right, now that you're picked up, I'm gonna run it into flush.
Roger.
My niece was pretty sure that I was actually at the bottom of the ocean when she was watching this earlier, and I let
And
her
I'm
think
at
that.
3/4
Yeah,
fly.
let her.
Roger.
She's very young. I'll tell her later. Oh, there's the kayak fish again.
Kayak fish.
Yeah, so we're
Yeah,
approaching this like little plateau, or a little summit over.
Oh, what's that?
Okay.
Sea urchin?
Oh, that is an urchin.
The round
Okay,
one?
Yeah.
Sorry, Marley.
Okay, flushed.
Looks like another sea star. Different one.
Cameras are back the way they're supposed to be, and I'm gonna come around to 040.
Roger.
Is the map that we're looking at, Ben, is it backscatter or what?
Mm-hmm.
What are we looking
On
at?
high pack.
On high pack.
Oh, that's just our let's see.
Is that our data?
No, actually, yeah, this is our data. She may have actually overlaid our backscatter.
Yeah, she did, because
Yeah.
earlier they were
It looks like, I mean,
they
it
were
looks like
wanting it
backscatter.
because it was very sedimented, so they wanted
right.
to make
Sure, sure.
or we're trying to get out of that.
Mm.
Yeah, I'm gonna swap out to the old map when we approach the
Yeah,
end
did
of
you
our
guys want to pass
coverage.
this around the van?
Neat little sponge down there.
Does anyone want some of these guys?
Not right now, but
What do I-
maybe
oh,
later.
Yeah.
Megan does.
Megan does.
I think I can eat those.
We on the data chart table. The chart table, not data chart.
So, back to the very important topic of our team name. I'm voting for Team Chocolate over Team Donut. Just let that be known.
This has been a very food-based dive.
Well, there was one of our viewers was asking how much coffee we drink, right? And I feel like this is more powered by chocolate candies.
Well, I mean, our watch does span dinner, so it's hard to not have food come up as a
Right,
topic.
sometimes you can smell breakfast, like the last hour.